This tuberculosis molecular bacterial load assay answers 3 key questions. 1. What is the magnitude of the patient's disease burden? 2. How does the patient disease burden respond to the antituberculosis medication? And 3. What is the relationship between disease burden and treatment response? This assay produces a quantitative result of the patient tuberculosis burden and measures how this burden changes with treatment in a short time. With modification, this methodology can be applied to other bacterial pathogens. We have already developed similar technology for diagnosis of non-tuberculous mycobacteria and bacteria associated with chronic obstructive pulmonary disease. When performing this technique for the first time, watch the video and practice the steps. It's very important to practice with samples that are not needed for patient diagnostic results. Visual demonstration is critical because it simplifies learning and mastering of steps, especially those parts of the method that are difficult to explain in text.
Start by preparing the cell cultures. On a clean lab bench or Class 1 cabin, harvest 1 milliliter aliquots of exponential phase Bacillus calmette-guerin or BCG culture into 15 milliliter plastic tubes and tightly close them. When preparing patient sputum samples, work in a well-ventilated space and follow the guidelines in the GL1 stop.